Welcome to The Explainer. Today, we're diving into a timeline that's honestly a little mind-blowing. We're going to see how centuries of scientific discovery mapped out the microscopic world and directly shaped the life-saving sterile techniques used in literally every single modern surgery today. It's a crazy journey full of brilliant minds, totally accidental discoveries, and a relentless pursuit of the truth.
But before we really get into it, just think about this for a second. How on earth do healthcare workers fight an enemy they cannot even see? To really grasp modern medicine, we have to raise the stakes. We have to look back at a time when the invisible world of microbes turned surgical rooms into these deadly, unexplained mysteries. Imagine going into surgery where the doctors didn't even wash their hands, just because they simply didn't know bacteria existed.
Well, today we handle this unseen enemy through a strict code called surgical conscience. It's basically an ethical armor rooted in the fundamental principle of agere crimo, which means the patient first. Think about it. It requires surgical techs to disclose any break in sterile technique, whether anyone else saw it happen or not. Because you can't see sterility, you can only assume it's there if the absolute best practices are followed, without exception.
So let's map out exactly how we built this foundation. We're going to trace a straight chronological journey, from the very first theories in 1546 to microscopes and cells in 1665, through the germ theory and hand washing of the 1800s, right up to modern virology in 1964. All right, kicking things off with the early pioneers, spanning the 1500s to the 1700s, the very first glimpses of the unseen.
Way back in 1546, a scholar named Girolamo Fracastoro lays down some brilliant theoretical groundwork. He theorized that unseen organisms were actually spreading disease through contact and even the air. Then, jump ahead to 1665, and Robert Hooke builds a compound microscope. He completely changes biology forever by giving us the basic unit of life, officially coining the term cell.
But what's really wild is the technology itself. In 1673, Anton von Leeuwenhoek used his famously simple microscope, literally placing the specimen right on the tip of a tiny pin, to become the very first human to ever observe invisible bacteria. He actually called these little guys animalcules. And in 1723, he submitted his incredibly accurate drawings of them to the Royal Society of London, which, quite rightly, earned him the title Father of Microbiology. Moving right along to the era of challenging dogma from the 1600s through the 1800s, the real battle for scientific truth. See, simply seeing these microbes wasn't enough. These pioneers had to fight completely unscientific entrenched beliefs like spontaneous generation, the idea that life just magically popped into existence from non-living stuff. Francesco Redi logically fought this. He proved that decaying meat in open jars developed maggots, while completely sealed jars remained clear, no maggots, but critics stubbornly kept the myth alive for a while, claiming the sealed jars just lacked fresh oxygen. Now, jump to the mid-1800s. Things get very real in maternity wards. Physician Ignaz Semmelweis notices a horrifying contrast. Medical students going straight from autopsies to delivering babies were causing massive maternal death rates. But the midwives? They had much lower infection rates because they were strictly washing their hands between patients. And here is the absolute kicker. Once Semmelweis forced physicians to use chlorinated hand hygiene, the maternal mortality rate plummeted to just over 1%. 1%. It was undeniable proof that hand washing saves lives. Right around this same era, Edward Jenner makes a colossal leap of his own. He noticed milkmaids were practically immune to deadly smallpox after catching the much milder cowpox. So he intentionally inoculates a young boy with cowpox, and boom, the boy shows total immunity when exposed to smallpox. Just like that, the father of vaccines successfully invents vaccination. Next up, the Golden Age, spanning the mid to late 1800s. This is all about revolutionizing modern surgery. Enter Louis Pasteur. He definitively crushes that old vital force nonsense once and for all. He formally establishes the germ theory of disease, linking microbes directly to illness. Oh, and he also figures out how to save food from spoilage through a low heating process we still use today, pasteurization. Total game changer. Naturally, Pasteur's work caught the eye of Joseph Lister, a surgeon who fully embraced this link between microbes and infection. Lister establishes the very first true aseptic techniques. He insisted on carbolic acid skin scrubs, surgical gowns, and boiling instruments. And that is exactly why we call him the father of modern surgery. Following Lister,
In 1876, Robert Koch brilliantly brings order to all this microscopic chaos. He develops Koch's postulates. These are strict experimental steps used to definitively link one specific microbe to one specific disease. He uses pure culture techniques with solid agar in petri dishes and the streak plate method to grow and identify bacteria. Koch essentially proved that different bacteria cause different infections.
Rolling into the 20th century, the stakes hit incredibly high numbers. In 1918, the devastating Spanish flu pandemic kills an estimated 20 to 50 million people, but humanity fights back, fast. In 1928, Alexander Fleming accidentally discovers the penicillin mold, and by 1945, the first influenza vaccines are successfully developed.
Which brings us to the modern era, in the 1900s, expanding the fight against pathogens.
Here we see a diverse, incredibly talented new generation of scientists making massive strides. Ruth Ella Moore, the first African-American to earn a PhD in bacteriology, conducts extensive research into treating tuberculosis. Elizabeth Bougie was part of the team that developed the antibiotic streptomycin to treat TB in 1944. And June Almeida? She pioneers groundbreaking electron microscope techniques.
To give you an idea of how far the tech had come by then, June Almeida actually gains global recognition because in 1964 she utilizes an electron microscope to become the very first person to ever identify a coronavirus.
So what does this all mean for us today? Well, all this historical research teaches us exactly where to intervene. By understanding every component of the chain of infection and the distinct ways it can be broken, every single healthcare worker knows how to stop a disease from spreading.
This entire legacy lives on in the CDC's standard precautions. We assume all body fluids are potentially infectious, period. We use strict hand hygiene, appropriate PPE, safe injection practices, and proper respiratory etiquette, regardless of a patient's known infection status.
These physical barriers, the face shields, the latex gloves, the non-absorbent gowns, and the N95 masks, these are the direct, modern manifestations of Lister and Semmelweis's early hygienic experiments. They are the frontline shields keeping both the healthcare worker and the patient completely safe.
So, armed with over 500 years of incredible scientific discovery, we have to look to the future and ask, how will the next generation of healthcare pioneers break the chain of tomorrow's invisible threats? Thanks for joining me on this explainer. Keep asking those big questions, and I'll catch you next time.